Right. So our team focused on a drug-eluting artificial bypass graft. I am Alyssa Webb.
I'm Julianna.
I'm Nicole Racca.
And I'm Emily Sansoucy.
So first, we wanted to focus on the patients who would need our device. So patients who require bypass surgery will have a blocked or narrowed artery that needs to be treated to restore the blood supply to their heart muscle. These patients can express the following symptoms: dizziness, chest pain, difficulty breathing, pale skin, sweating, arrhythmias, and nausea. So why is it important? More than 500,000 bypass grafts are performed each year, and this number is expected to increase with the aging population. About 40% of vein grafts fail within 18 months. Patients with obstructive coronary artery disease can benefit from bypass grafts, as it's a form of treatment for this disease due to the buildup and blockages in plaque. And most needs for an artificial grafts concern the lack of or unsuited veins.
Bypass surgery is more invasive surgery than the use of that, and it will require a longer recovery time or will have a higher rate of infection. We are looking at in hospital by what it was easy.
The coronary arteries are the transport vessels for the blood to the heart. As mentioned, OCID is characterized by the buildup of lipids on the arterial walls. The pathology is initiated when a piece of cholesterol becomes lodged in the arterial wall, which causes blood cells to target the area. As the cholesterol is encapsulated, inflammation occurs, and ultimately this results in lipid accumulation. There are three main categories of obstructive coronary artery disease treatment. They're oral drug therapies, angioplasty with stents, and coronary artery bypass surgery. Advantages of the first two treatments are that they are minimally invasive. Some disadvantages of drug therapies are the side effects of such as muscle pain or increased bleeding, and a limitation of stents is that it's not an appropriate treatment for patients with diabetes. A distinction for our device is that it's not intended to serve as a replacement for oral drug therapy or stents, but is instead intended for patients that coronary artery bypass graft surgery is the best treatment plan. There are no artificial bypass grafts approved by the FDA, but there are a few in development. Some examples are a hybrid bypass graft, a tissue engineered conduit, and a drug-eluting vascular prosthesis. All of these grafts use a flexible polymer, and the first two use tissue engineering techniques. A drawback of artificial grafts is a high rate of stenosis, and again, a lack of FDA approval. So we are proposing an artificial bypass graft that is made of PCL and is seated with the patient's endothelial and smooth muscle cells. The device will also incorporate the release of two drugs, Pravachol, an anti-hyperlipidemic drug, and Durlaza, an anti-inflammatory drug, that will help reduce the buildup of lipids and prevent inflammation. Studies involving the combination of these drugs have shown a decrease in the risk of heart attacks and strokes, and that the side effects were not increased. We'll use two-stage seeding where the cells are harvested from the patient from a vein or artery and cultured in the lab before being seeded onto the device to improve the biocompatibility and decrease the risk of failure. Although these cells are being gathered from a diseased patient, the disease does not typically affect the functionality of these cells, but that is still a potential limitation. To ensure that the cells properly coat the interior of the device, it can be subject to preconditioning, which is when the device is exposed to an in vitro pulsive flow and pressure, and it has been shown to promote cell proliferation and tissue formation. If upon testing the cells don't properly adhere to the device, we could explore the use of attachment factors such as collagen or fibronectin. Our clientele estimation is roughly 200,000 patients per year.
So shown here is the breakdown of our cost for a device, and there's a breakdown per individual material. And when factoring in all these individual costs, along with additional lab and preliminary cell culture costs, our device is approximately $36,160.
He hardly will be hardly irregular, hardly any higher order with OCAD you need by that hardly.
So our synthetic graft is preferable to the traditional autograft procedure as it does not require surgical removal of a healthy vessel from the patient, and it removes complications or infection at the donor site, and its targeted drug delivery mechanism aims to reduce infection and stenosis. If our solution is not implemented, CAPG surgery will continue to be hindered by these shortcomings, and some disadvantages to our solution is the idea that harvesting, cell seeding, and experimentation for a device will require several weeks of culturing, causing a delay in manufacturing time. And also the tissue growth following the degradation of PCL will not be insufficient condition to withstand the forces of the human body. But considering cell regrowth, the new graft may be insufficient condition within 2 to 6 months before the PCL degrades. Another limitation to the device is that there is currently limited research regarding the targeted release of Prevacol and Durlaza, which would then require further research on the team's behalf before the manufacturing process is commenced.
For engineering criteria and tissue responses, our device needs to withstand the forces applied by the pulsive flow of blood. In the coronary artery, this responds to 1.4 pascals of shear stress and 5 to 10% cyclic stretch. For tissue interactions, our device should support tissue regrowth of the artery. Ideally, there should be no negative effects on the blood that the device is in contact with.
So each test will be performed 5 times per graft style, and our graft styles will include our synthetic graft and 2 naturally derived controls. These controls will include the human saphenous vein and the internal mammary artery.
I know how we want him in the road where it really what he would really use it for. Let me but a higher row of he be permanent while maintaining a dolly calendar. We will measure at rock point on the floor. Let him have it. The FDA has any government about and the government will be on Canada for the AAMI we wanted 1994 what you really on here.
For hemocompatibility tests, the international standard states that for synthetic or tissue vascular grafts, tests determining the device's thrombogenic and hematologic properties are required, and they will also test the effect at blood coagulation. The 3 ASTM standards are shown on this slide. For all of these experiments, 3 trials we conducted. For the thrombosis test, black rubber will be used as a positive control, and high-density polyethylene will be used as the negative control. Platelet and leukocyte count is determined after the samples are exposed to fresh human blood. A large decrease in the cell population of the blood after testing would indicate that the cells are trapped in thrombi. The hematology standard determines the hemolytic properties of the materials. The time frame for the extract test is 15 minutes, and for the direct contact, it's 3 hours. Controls are washburn and rubber and polyethylene. For the coagulation test, it determines the time it takes for plasma that has been exposed to the sample to clot upon exposure to a suspension of phospholipid particles and calcium chloride. The controls are glass tips or beads and high-density polyethylene. A limitation of these tests is that they don't account for the pulsive flow of blood, which could have an impact on the outcomes.
So to determine the cytotoxicity properties of our device, along with determining the upper and lower limits of the individual drugs and the combination of the two, we'll be conducting hybrid-lytic assays. And these tests will allow us to determine the cytotoxicity of the eluted drugs at varying potencies and the reaction of the seeded cells to these drugs. Hybrid-lytic assays use fluorescent tag proteins to represent the viability of these cells in these conditions. Before the testing process is commenced, the cells will be cultured in sterile laboratory conditions and incubated. These drugs will include the drugs Durlaza and Provocol will be serially diluted for 6 repeat dilutions and 12 points into the plate starting at 162.5 milligrams per well for Durlaza and 40 milligrams per well for Provocol. The 23 column of the 384 well plate will be just cells for control, and the 24 column will contain nothing for background control. These cells will then be seeded into 4 384 well plates at 10,000 cells per well, and these cells will then be incubated in the conditions for 6 hours before the hybrid-lytic detection reagent and substrate will be added. And following the addition of the solution, the plates will then endure another incubation for 20 minutes or an hour. These plates will then be read on high-throughput imaging machines underneath the hybrid-lytic assay protocol to take count regarding the level of luminescence in each well of the plate. Later, the raw data will then be analyzed through Graft Prism to further determine the limits of each drug compound and the ideal IC50, which is the potency of the inhibitory substance. The incubation time of the assay may vary and be extended to 18 or 24 hours depending on the initial noise found in the raw data readings. And with longer incubation time, the amount of the noise found in the readings may decrease to produce a smoother viability curve. This assay is also very similar to a cell titer glow assay, which may be used to achieve similar results to find the cell viability underneath these conditions with the chosen drug compounds.
Since there is some overlap with our other tests and assays, biocompatibility can generally fall under this one umbrella ASTM standard. This standard is a general practice and focuses on a wide variety of tests and involves a biocompatibility matrix that should be completed, and some of these requirements may not necessarily specify certain tests. These tests that are performed involve testing the device to its endpoint, which in our case would be when PCl fully degrades.
So by completing in vivo testing, the team is looking to determine the local and systemic inflammatory responses the device invokes, along with the wound healing and tissue interactions and processes that the device undergoes. To complete these tests, the team will look into conducting short-term and long-term in vivo testing to fully understand the implementation responses. In the body. For short-term testing purposes, of approximately 60 days, rabbit or rat implementation has been chosen. Rabbits were chosen for the high similarities found in their vascular system in comparison to humans. Unfortunately, though, because of their size and stature, rabbits are a difficult specimen for animal testing because the surgical processes of implementation is increasingly difficult in comparison to other small animals that are used for short-term testings. Rats were also determined as a good specimen for animal testing because of easy implementation, but the vascular inflammatory system of a rat are not as comparable to that of a human. Rats have a lack of macrophages like M1 and M2 in comparison to the human system, which further alters the tissue interaction and wound healing response, requiring further testing to be done to determine responses in the human body. For long-term testing, sheep, porcine, or goat caprine animals were chosen for ideal conditions to analyze the inflammatory and immune responses to the device, along with long-term tissue interactions and reepithelialization in comparison to humans. Sheep and goats were chosen as an ideal long-term, larger specimen for implementation because their larger stature and larger bodies allow for easier access for implementation of the artery. They are also ideal for testing because the 4 to 6 millimeter in diameter blood thrombogenic mechanisms, which are highly similar in stature to those of humans. Sheep and goats are also quickly maturing animals and are highly cost-effective solutions to determine the long-term implementation of these devices in comparison to the cardiovascular physiology with humans. Porcine and ovine implementation models are excellent choices for preclinical testing that can be used for short-term patents or long-term remodeling of tissue-engineered vascular grafts.
So as PCL has a low melting point, many traditional sterilization techniques aren't suitable, but gamma irradiation and ethylene oxide sterilization are. For gamma irradiation, a dosage of 29 kilograms has been shown to be successful with no changes in morphology of the PCL. And ethylene oxide sterilization is also suitable as it doesn't affect the degradation rate or the tensile modulus. However, the procedure is of high cost and ethylene oxide is a known carcinogen. So ideally, our device will be sterilized through gamma irradiation.